Welcome back once again to this educational channel called Learn Bio with Janet. Today we'll be discussing the structure of RNA or ribonucleic acid, and there will be also a summary about the structure of DNA, deoxyribonucleic acid, as we have studied it in the first video. Finally, we'll make some interesting comparisons between RNA and DNA from various aspects. Let us get started. I've thought up two useful acronyms that you can use to remember the 4 bases of DNA and the 4 bases of RNA. The 4 bases of DNA are adenine, thymine, guanine, and cytosine. Okay? And their symbols are ATGC. So there's no uracil in the DNA molecule. Okay, the thymine replaces the uracil. So remember, dine at the garden cafe. D is for DNA. Okay, and A is for adenine, T is for thymine, G for guanine, and C for cytosine. So after a vaccine has been found for this virus, then we can dine freely at the garden cafe. Okay. Now, secondly, let's look at the 4 bases of RNA. Uracil, adenine, guanine, and cytosine. Uracil in RNA replaces the thymine in DNA. Okay. So remember, round up all greedy crows. R is for RNA, U for uracil, A for adenine, G for guanine, and C for cytosine. Simple as that. Okay, so RUAGC for RNA. Now, if you don't want, you can put round up angry greedy crows. Crows are very angry sometimes, and they are greedy. I remember in the school canteen, they will fly down from the skies and try to eat the leftover food before it's collected back, okay, by the stalls. So round them all up, all the greedy crows, before you dine at the garden cafe. Okay, I hope you will be able to use these acronyms that are very useful. It's up to you. If you feel that you don't want to use it, then you don't have to. But I would say that these are quite good acronyms. Only use the first letters of each sentence. And you can repeat these 2 phrases many times in order to remember. Here is another set of interesting notes which I have created to help you understand and remember the structure of DNA and RNA more easily, and also to compare them. So this shows you how the DNA molecule is built up. You see, the components here are the simplest ones. They are the phosphate group, sugar, and base, which join together during the process of condensation. Okay, they combine together during condensation to form a nucleotide. Next, when many nucleotides join together, now I just write the short form of the word. Of course, you can't spell it like that in the exam, but it's okay because these are short notes, right? So many nucleotides join together to form a polynucleotide chain. Okay, like one of these, like a pink chain here, or strand. And when there are 2 polynucleotide strands that join together, pink one and the blue one, okay, they are joined at the bases by using hydrogen bonds. And when there is a twist to that structure, then you get the double helix structure of DNA, as shown here, where the 2 polynucleotide strands, the pink one and the blue one, are intertwined. It means they coil around each other, right, to form the double helix. Then for the structure of RNA, is simpler. A phosphate group, a sugar, a pentose sugar, and a base combines during condensation to form one nucleotide, which is the basic unit of both RNA and DNA. then many nucleotides join together, as for DNA, and form one polynucleotide strand or chain times 1. So there's 1, and that's all. There's no combination of 2 strands. Okay, so RNA here is made up of 1 strand, as you can see here. 1 strand of polynucleotide compared to DNA that has 2 strands of polynucleotide. And secondly, the strand of RNA is shorter than the strand of DNA, much shorter. So if you find that this is useful, you can copy it down in your notebook. And during revision, it will help you to remember and understand better the structure of DNA and RNA, and to even compare them. Let us discuss the structure of DNA as a revision, right? So the summary of the structure of DNA. The basic unit of DNA are the nucleotides, as shown here. Each nucleotide is made up of 3 components, the P, S, and B. P for phosphate, then we have the pentose sugar, which is deoxyribose for DNA, and a nitrogenous base such as adenine, thymine, guanine, or cytosine, just 1 base. So remember the acronym, please stay back, PSP. Now, many nucleotides are joined together to form a long polynucleotide strand. So this nucleotide is joined to the next one and the next one and the next one to form a long polynucleotide strand, where the pentose sugar of the first nucleotide will be joined to the phosphate group of the second nucleotide, and so forth. Okay, so we will get 1 strand of polynucleotide, like just 1 of these, after the nucleotides are bonded together. Then 2 different strands, like shown here, this strand and this strand. 2 different strands of polynucleotides are bonded together by hydrogen bonds in the center here, between their bases. Okay, so these colored parts show a pair of bases. One is from the first strand and the other one is from the second strand. So there are hydrogen bonds between their bases to bond the 2 strands together, bind them together, and to form the DNA molecule. The 2 strands are antiparallel, that means they face opposite directions. One will have the sugar facing downwards and the other one the sugar facing upwards. Okay, and are intertwined. Intertwined means they are coiled around each other, like this one, coiled around, in opposite directions to form a double helix. So this is the name of the structure of DNA, a double helix. Next, the nitrogenous bases on one polynucleotide strand are matched with their complementary bases on the other strand. So here, if you have, for example, in this picture, you have adenine, the base adenine A, then it will always be paired with thymine, T, because these 2, adenine and thymine, are complementary bases. So they will pair with each other. Whereas guanine always pairs with cytosine. Okay, also in the center of the double helix. And that's the structure of DNA. Now let's discuss the structure of RNA. RNA is a single-stranded nucleic acid used in protein synthesis. Only 1 strand. Okay, location found in the nucleus, cytoplasm, and ribosomes. Types, there are 3 types of RNA. The first type is called messenger RNA. So, as its name implies, it carries genetic information from DNA to the ribosome. First, it will copy the genetic information from DNA in the nucleus. Then after it has copied the information, it will come out from the nucleus into the cytoplasm to the ribosome. And on the ribosome, it will sit there, and then the genetic information is translated. Okay, it will be made known, it will be read. Okay, we'll come to that later. So ribosomal RNA, as its name implies, is in the ribosome. This is called rRNA, and it's used to make the ribosome. It's a component of every ribosome. Transfer RNA or tRNA transfers amino acids to ribosomes. Now, don't worry about the function of these three, as we'll be studying them in the next video, okay, when we talk about the synthesis of proteins. Okay, function of RNA. So here is one function of messenger RNA. Okay, the rest are not mentioned. Messenger RNA copies DNA's genetic code in the nucleus, as we said before. Because DNA molecule is very big, it doesn't come out from the nucleus, and it's too precious to be left in the cytoplasm. It must be kept inside the nucleus, like in a warehouse. And the messenger RNA will go there and copy the code from DNA, and then it will come out through the nuclear pore and carry the code to the ribosome, the genetic code for the production of one protein. Okay, it will carry it to the ribosome in the cytoplasm to direct the synthesis of proteins on the ribosome. So here we see a strand of RNA, a short strand. It will go to the ribosome like this, and it will sit on the ribosome here, and the ribosome will read the genetic code to find out what amino acids have to be assembled or collected or joined together to form that particular protein. By reading the code, it will determine the types of amino acids that have to be joined together to form the protein, because there are many different types of amino acids that are used to synthesize the proteins. Okay, let's discuss this learning outcome. Describe the structure of RNA or ribonucleic acid. Now, this can be asked as a question in the exam too. Just like we have described the structure of DNA or deoxyribonucleic acid, so now here we have to describe the structure of RNA. Now, basically the RNA molecule is a polymer made up of monomers or basic units called nucleotides. Now, that's similar to DNA, right? So RNA is also a polymer like DNA, made up of this basic unit of phosphate group, sugar and base. And this basic unit that has these three components is called the nucleotide. So the components of a nucleotide are the phosphate group, pentose sugar, but in this case the sugar is not deoxyribose, it is ribose sugar. DNA has deoxyribose sugar, okay, but RNA doesn't have deoxyribose sugar. Instead, it has ribose sugar. There's a slight difference in the molecular structure and formula, in the formula of the two pentose sugars. Okay, so RNA has the sugar called ribose, and lastly, the sugar is joined to a base, a nitrogenous base that contains nitrogen. Now, what are the four nitrogenous bases that can be found in the RNA molecule? Base can be adenine, guanine, cytosine, and uracil. So this is a new base that we have not studied before. Uracil replaces thymine. So uracil in RNA replaces thymine in DNA. So there is no thymine in RNA, only uracil to replace the thymine. Okay, so one nitrogenous base such as adenine or A, uracil or U, no T, no thymine, no T, guanine G, and cytosine C. So from here we already found two differences between DNA and RNA. DNA has the pentose sugar called deoxyribose, but RNA has the pentose sugar called ribose. DNA has the base called thymine, but RNA does not. RNA has the base called uracil, represented by a U. Okay, further explanations on the structure of RNA as follows. Thymine in DNA is replaced by uracil in RNA. And D, many nucleotides will join together to form a long polynucleotide strand or chain called RNA. Okay, like this shown here. Now, the RNA molecule is a single strand of polynucleotides. Normally, it's about a few thousands. It has a few thousand bases, a few thousand nucleotides, all joined together. Okay. Now, why is it that the RNA molecule is shorter than DNA? So, RNA is shorter as it is produced from only a small section of the DNA. In fact, RNA is formed when the code for the production of one protein is copied from DNA. And the code for the production of one protein is called the gene. Okay, so which is only one gene with information to produce only one protein. Whereas DNA stores many genes. Okay, one strand of DNA will store many genes, thousands of genes. Then lastly, we can mention what the elements of RNA are. They are carbon, hydrogen, oxygen, nitrogen, and phosphorus, just like for DNA, and that is the acronym CHOMP, C-H-O-M-P. If you are asked to draw the molecule of RNA, you can draw it in this form using the nucleotides, just like for DNA. So the first nucleotide here, consisting of the pentose sugar phosphate group and nitrogenous base, is linked to the next nucleotide and then to the next one, where the pentose sugar of the first nucleotide will bond with the phosphate group of the second nucleotide, and the pentose sugar of the second nucleotide bonds to the phosphate group of the third nucleotide, and so forth, as we have explained for DNA. Okay, and here is the structure that we've discussed just now. To compare the two, right, they are the same. The base will project out from the sugar phosphate group backbone. Okay, so this one is still called the sugar phosphate backbone, like for DNA, and the bases project out from there. So RNA is a single strand of nucleotides, seen here or here, and the base thymine is replaced with uracil. So don't ever draw label the base as T. Okay, for RNA it should be U. There's no T or thymine in RNA. Here's some advanced knowledge about the structure of the sugars, the deoxyribose and ribose, thymine and uracil, to show the differences between DNA and RNA. Now, you may not need to memorize these structures of these molecules, okay? Just understand the differences. Deoxyribose sugar looks like this, and ribose sugar is this. The difference is in this group here, or atom here. Deoxyribose has a hydrogen atom here, but ribose has a hydroxyl group here. So the name deoxyribose is derived from this difference, because it does not have the oxygen atom, as in ribose. It's called deoxy, meaning without the oxygen atom. Ribose molecule without the oxygen atom. So deoxyribose. Okay, and then the base thymine and uracil. Thymine is found in DNA, uracil in RNA. They are very similar except for the difference here. CH3 group is found here, and this one is only a hydrogen atom. Okay, so these are two differences between DNA and RNA. Here are some very good short notes to study the differences and explain the differences between DNA and RNA. They are short and sweet, so it's easy to remember. You can copy this down if you want, and this question can be tested in the exam. So here on the left is DNA and on the right is RNA. Okay, so what are the differences? Firstly, the bases. DNA contains the base thymine and also adenine, cytosine, and guanine. There's no uracil. RNA contains the bases uracil, adenine, cytosine, and guanine, and there's no thymine. Okay, so the thymine in DNA is replaced with the uracil in RNA. Length. DNA is very long because it stores a lot of genes for the synthesis of various types of proteins. But RNA is shorter because it only copies one gene for the production of one protein, and it copies this from the DNA. Now sugar, the pentose sugar in DNA is deoxyribose, but in RNA is ribose. Strand, DNA consists of 2 strands of polynucleotides, but RNA consists of only 1 strand of polynucleotides. Function, DNA stores genetic information that is inherited, that can be inherited or passed down to the offspring, from the parents to the offspring. So inherited means boleh diwarisi, can be passed down. Now, the genetic information is stored in the sequence of bases here, and we will study that in the next video. So this information can be passed down to the offspring through the sperm and the ovum from the parents. For RNA, its job is to copy the genetic code for the synthesis of one protein from DNA, and then it will transfer this genetic information for protein synthesis by coming out from the nucleus and into the cytoplasm to the ribosomes. And the ribosomes get the genetic information to synthesize the proteins. Okay? Again, we will talk about synthesis of proteins in the next video, and it will become clearer to you. Now, location of DNA is that it is located in the nucleus and mitochondria, okay, these 2 organelles of the cells. Whereas RNA is located in the nucleus, cytoplasm, and in the ribosome. Okay, it is used to as a component for the synthesis of the ribosome. Now, acronym. You can use this acronym to remember the various aspects for differences between RNA and DNA, and it's called blast flow. Okay, let's see. Now the aspects are here. So B is for the bases, L is for the length, LE is for length, S is for sugar, and S is for strand, blast. And then flow, F is for function, and LO is for location. So it's as simple as that. If you memorize this acronym and you are asked this question in the essay section or structured question section, then you can easily recall, more easily recall the various aspects to explain. Let us now discuss a forecast exam question, which is very likely in the coming years. Okay, compare the DNA and RNA molecule, 10 marks. Now this one is an essay question because 10 marks are allocated for it, but it may come out as a structured question where less marks are allocated, like 2 to 4 marks. Okay, and even for objective questions, you can come up the similarities and differences between DNA and RNA. Okay, so if you're asked to write an essay answer on the comparison between DNA and RNA, you must state both the similarities and the differences. If you have 5 similarities, you should have another 5 differences, because each similarity is 1 mark and each difference is 1 mark. 5 similarities, 5 marks. Okay, 5 differences, 5 marks. So together, 10 marks. Okay, so you have a mixture of similarities and differences. Let's begin. Now, let's try to challenge ourselves to give the right or the correct terms for these answers. Both are what type of acids? Wow, that's a very loud applause. Okay, both are nucleic acids, which carry what type of material? Yes, genetic material. Now, both DNA and RNA contain the bases. There are 3 bases that are the same, okay, for both RNA and DNA. A, C, and G. A for adenine, C for cytosine, and G for guanine. Adenine, guanine, cytosine. Both DNA and RNA are polymers which have the same basic units called nucleotides. Now the backbone of both molecules is, what two components of the nucleotides form the backbone of the RNA and DNA molecule? The sugar phosphate group. Good. Now both are formed from the elements. There are 5 elements, that form the DNA and RNA molecule. And the short form is CHOMP. Remember CHOMP? C for carbon, H for hydrogen. Then we have O, N, and P. Oxygen, nitrogen, and phosphorus. CHOMP. Both are found in which part of the cell? Yes, the nucleus. And lastly, both are involved in the synthesis of a substance in the cell. What is the substance? Good, proteins. Right, so now let's have a look at how the 2 nucleic acids are involved in the synthesis of proteins in the cell. DNA in the nucleus provides the genetic information about protein synthesis. So DNA is like a storehouse of genetic information for various types of proteins. And then the RNA will copy the information for the production of just one protein. That means it copies one gene with the information for production of one protein, and then it brings it out to the ribosomes, from the nucleus to the ribosomes, which are in the cytoplasm. And the ribosome will carry out the synthesis of proteins by using this genetic code. So both DNA and RNA work together to synthesize the proteins, to help synthesize the proteins. Now let's discuss the differences between DNA and RNA. For DNA, its pentose sugar is deoxyribose, and for RNA, its pentose sugar is ribose. DNA is how many stranded? Double. Consists of 2 strands of polynucleotides, and it forms a double helix. RNA, it is single-stranded, and it usually doesn't form a double helix. The base adenine in DNA pairs with the base thymine. There's no uracil. But in RNA, the base adenine pairs with uracil, and there's no thymine. DNA, its length is longer or shorter? Yes, longer. And for RNA, its length is shorter or short. Now, DNA is located in which part of the cell? 2 organelles, in 2 organelles. Nucleus and mitochondria. In RNA, and RNA is located in where? Where's RNA located? Nucleus, cytoplasm, and ribosome. Now, DNA is self-replicating. It can duplicate itself, especially during cell division, so that the new cells will also have a copy of the same DNA. So it can replicate itself. Or the chromosomes will duplicate themselves, and in the process, the RNA is duplicated too. Now, for RNA, it cannot self-replicate. It is synthesized from DNA by copying the code on DNA. So RNA is like, well, in inverted commas, the daughter of DNA. It is from DNA. But whereas DNA is double-stranded, RNA is single-stranded. Now, DNA is involved in heredity or inheritance by storing genetic information and passing it on down to the offspring, from parents to offspring, and then from the offspring to the offspring's children, and so forth. So heredity or inheritance, in Chinese we call it yichuan, and in Malay is called pewarisan. Terlibat dalam pewarisan. That means the passing down of the characteristics of the parent or the genes of the parent to the offspring or the children. But RNA is not involved in heredity. It is involved mainly in the synthesis of proteins in the cells. Okay, so in the next lesson we'll study about the 3 types of RNA and how they're involved to synthesize the proteins. It's quite an interesting story. Now that ends the video for this lesson. That ends the lesson, and I'll see you in the next video. I'll be finishing up this topic on synthesis of proteins. Okay, so stay tuned for that, as that needs some explanation to help students understand. And finally, share, comment, like and subscribe. Thank you for watching, and see you again in the next video.